Hey all, Azim here. We're going to take a look at two different models that show the kidney, but more specifically, the nephron level of the kidney, which you can only really see microscopically. So these models are representing that. Let's start with our zoomed out look at the kidney. Here we have a whole kidney. This is showing the left kidney because we have a frontal cut. You can see the ureter here. So this is the anterior side, but cut in half. Fibrous capsule is what's protecting the kidney on the outside. It's not that thick, it's just a fibrous capsule. This region here, that's cortex. That's the cortex of the kidney, the renal cortex. Deeper in, that's the renal medulla. You can see what's part of the renal medulla, these triangle shapes. Those are renal pyramids. And then in between the renal pyramids, those are renal columns. The columns have blood vessels going in and out to the cortex. The nephrons are found throughout the cortex and medulla. They always span both of them. So shown here is one nephron. You can see the beginning of it right here. There's the beginning where that circle is, and then it goes down into the medulla, it comes back up into the cortex, goes back down into the medulla, and then ends at the renal papilla, the tip of the renal pyramid. And then urine exits into the minor calyx, major calyx, renal pelvis, ureter. So let's take a closer look at one of these nephrons. So these tubes are called nephrons. So they're the functional unit of the kidney. They are how we are able to filter our blood and reabsorb important nutrients and secrete more wastes and then eventually excrete what we don't need, excess stuff and waste. So shown here are two nephrons. There's one nephron right here, starting here. I'm going to trace it. This is the Bowman's capsule, the glomerular capsule, followed by the proximal convoluted tubule. Convoluted just means it's really twisted. You can see how twisty it is. Proximal convoluted tubule, descending limb of the loop of Henle, which goes into the medulla. This half is medulla. The loop of Henle goes into the medulla, then it comes back up. This is the ascending limb of the loop of Henle. And then the distal convoluted tubule. It's distal because it's farther away from the Bowman's capsule in terms of our path. The distal convoluted tubule actually goes right in between here, and it'll be more apparent on this next part. Distal convoluted tubule. Here we have a connecting tubule. This connecting tubule connects several nephrons, like this one and this one. There would be one here, one cut off here. Connecting tubules connect to the major collecting duct. This is a collecting duct. Once we get to the papilla, now it's a papillary duct. And then the urine will exit out here. Blood vessels that we see here. This blood vessel going into the Bowman's capsule, this is the afferent with an A, afferent arteriole. The blood vessels in here are capillaries. They're fenestrated capillaries called the glomerulus or glomerular capillaries. Glomerulus means a ball-shaped. And then when the blood exits, this is not a vein, it's another arteriole because we have a portal system here. This is the efferent with an E, efferent arteriole. There would be peritubular capillaries that would go along the proximal and distal convoluted tubule. That's shown better here. These are peritubular capillaries. So another set of capillaries, as well as capillaries that extend alongside the loop of Henle. That's called the vasa recta, V-A-S-A-R-E-C-T-A, vasa recta. So once again, afferent arteriole, glomerular capillaries, efferent arteriole, peritubular capillaries, and vasa recta, and then we exit out in this blue one to the renal vein. It'll branch more, but I just need you to know the term renal vein. We're going to zoom in on this portion of the nephron. That's right here. Actually going to rotate it because it might make it simpler to understand. This is representing the afferent arteriole with an A. Afferent arteriole brings blood in, and then we branch to capillaries. These capillaries are the glomerular capillaries. They're surrounded by these special cells, shown here in blue, called podocytes. Podocytes wrap around and help filter stuff as it leaks out of these glomerular capillaries. This is a lumen space, and this structure made of simple squamous epithelium is the Bowman's capsule, the glomerular capsule. So this is catching everything that's been filtered out of the glomerular capillaries, catches it in the Bowman's capsule. This is the efferent arteriole. Efferent arteriole, the blood exits here. So these capillaries are not for exchanging nutrients to feed these cells per se. Their main function here, is to filter the blood. Things are getting filtered out here. These are fenestrated capillaries, the glomerulus. This is the proximal convoluted tubule. We're transitioning from simple squamous of the Bowman's capsule to simple cuboidal, and these simple cuboidal epithelial cells have microvilli. The reason is we want to increase the absorption of nutrients like glucose and salts from the lumen here to the paratubular capillaries, which would be over here. You would go Bowman's capsule, proximal convoluted tubule, descending limb, ascending limb. The distal convoluted tubule would come back up around here, right in between the afferent and efferent arterioles. So that's how twisted this tube is. The distal convoluted tubule is shown here, and it's cut off. The distal convoluted tubule is also simple cuboidal, but no microvilli. We do get some absorption here, but we also get secretion. That means stuff from the distal convoluted tubule in the lumen will get, stuff from the paratubular capillaries will get secreted into the distal convoluted tubule. For instance, if you take if you have excess waste and it didn't get filtered out here, you get a second chance to filter it out here, except it's more targeted filtration. It's not filtration because it's targeted, so that's why it's called secretion. You have specific protein transporters in the membranes to transport those solutes to the lumen here. Another added benefit of having the distal convoluted tubule come right by the afferent and efferent arterioles is that now we can sense, the afferent arteriole can sense what is our filtration rate, what is our glomerular filtration rate. There are cells here, there are special cells here in the macula densa. These are macula densa cells that sense that glomerular filtration rate, and we can adjust the size of this arteriole to increase or decrease the rate as needed. We want to keep glomerular filtration rate constant. That's something you'll learn in physiology. We want to keep glomerular filtration rate constant, so by monitoring what's going on in the distal convoluted tubule, the afferent arteriole can do that, these macula densa cells of the afferent arteriole. This whole thing then, since we have the distal convoluted tubule and these arterioles that are sensing what's going on here, this is known as the juxtaglomerular complex. It's next to the glomerulus. Juxtaglomerular complex. I believe that's it for this model. Let's take a look at another one. All right, let's take a look. Where should we start? Let's start here. Oops. All right. So this is showing a whole kidney. Capsule, cortex, renal pyramids of the medulla, renal columns of the medulla, minor calyx, major calyx, renal pelvis, ureter. What this is showing in the background, so if you can ignore this for a second, when you look at the histology which we will look in another video. When you look at the histology of the kidney, there is a distinction between the cortex and medulla. The cortex tend to have smaller tubes, smaller lumens, because as these tubes bend and twist, when you cut them, you're more likely to get a circular shape. But by the time we get to the pyramids, you just get really long tubes that are going to empty out into the minor calyx. And so you can see these longer tubes here. They're both simple cuboidal, mostly. Simple cuboidal cells are just the size of the lumen, the shape of the lumen. It tends to be longer in the medulla. Here, we're looking at one pyramid. This is a single renal pyramid. Capsule, cortex, medulla, renal papilla. There are several nephrons shown here. There are three nephrons shown in this renal pyramid. Let's follow one of them. Shown in yellow is a Bowman's capsule. If we were to cut it open, and that's what's shown here, you could see the glomerular capillaries. So these are efferent arterioles going in. Bowman's capsule, proximal convoluted tubule. It's convoluted. Descending limb of the loop of Henle. The descending limb gets thin, so it gets to be simple squamous at this deep part of the loop of Henle, the descending limb of the loop of Henle. By being thin, it will absorb water very well, but it's made to not absorb salts. The ascending limb is made to absorb salts, but not water. And the reason why we have this dip down into the medulla, we're increasing surface area of the tube, and for other mechanisms of what's going on here with these cells and their transporters, we can maximize water reabsorption. The kidneys, not only do they filter out waste, but another super important function of the kidney is to conserve water. By having a long loop of Henle going down deep into the medulla, we can conserve more water. Some loops of Henle are shorter, some are longer. There's ones that are closer. The Bowman's capsule is closer to the medulla. There's some that are farther away, but they all basically have the same parts and same functions for the most part. Descending limb of loop of Henle, ascending limb, it's simple cuboidal again. Distal convoluted tubule. I said that the distal convoluted tubule goes in between the afferent and efferent arterioles. We'll take a look at that again in another angle of this model. Distal convoluted tubule, and then it connects, this is the connecting tubule, to the collecting duct. You have one connecting tubule here from one nephron. This one is over here. Here's another connecting tubule to from another nephron. Here's another connecting tubule from another nephron. You can trace it: Bowman's capsule, proximal, descending limb, ascending limb, distal, connecting tubule, collecting duct. Bowman's capsule, proximal convoluted tubule, descending limb, ascending limb, distal convoluted tubule, connecting tubule, collecting duct. Collecting duct is simple cuboidal. This is where the antidiuretic hormone acts if you want to absorb even more water. This is where aldosterone will act if you want to absorb sodium to modify your blood pressure and also salt levels. We then transition to the papillary duct in the papilla. The papillary duct is simple columnar. So we had simple cuboidal for most of this, simple squamous in the Bowman's capsule, simple squamous in this descending limb, but simple cuboidal in most, and then simple columnar in this last part, the papillary duct. And that exits to the minor calyx. This one is great for showing all those blood vessels. So let me show you the nephron real quick. Bowman's capsule, proximal convoluted tubule, descending limb gets thinner here, ascending limb gets thicker, distal convoluted tubule, connecting tubule, collecting duct, and then papillary duct will be farther down. Into the Bowman's capsule, we've got our afferent arteriole. All these capillaries around are Peritubular capillaries, if they're in the medulla, it's the vasa recta. So vasa recta is absorbing stuff from the loop of Henle. Peritubular capillaries are absorbing stuff from the proximal and distal convoluted tubules. And you can notice that the distal convoluted tubule is going in between the afferent and afferent arterioles. Here we're singling out one nephron and looking more at different parts of it. You can see the different types of epithelium, like on the thinnest part of the descending limb and wrapping around the loop, it's simple squamous. Most places it's simple cuboidal. The proximal convoluted tubule has microvilli. Here's the Bowman's capsule, which is encapsulating the glomerulus, the fenestrated capillaries. I suppose this would be the afferent arteriole. I think this is cut away because they want to show you the macula densa. Afferent arteriole, blood goes in, goes around, comes out. Here's the afferent arteriole. So this is the juxtaglomerular complex. It's all just twisted together. It's not a very neat looking thing. And then going in closer, here we have our afferent arteriole, glomerulus, with podocytes wrapping around the glomerular capillaries to aid with filtration. Efferent arteriole, Bowman's capsule with its simple squamous, proximal convoluted tubule with its microvilli, distal convoluted tubule, forming the juxtaglomerular complex. You can see the microvilli here. Focuses, there we go. Microvilli of the proximal convoluted tubule, just simple cuboidal with no microvilli, and the distal convoluted tubule. All right, I believe that's it. There's a lot here, a lot of important parts of the kidney, parts of the nephron that are vital for filtering, reabsorbing, secreting, and excreting, and then blood vessels that go along with it. To repeat what the blood vessels are: renal artery, which branches a bunch. We eventually branch to a afferent arteriole, glomerular capillaries, efferent arteriole, peritubular capillaries, and vasa recta, and then we exit out, branching back to a renal vein. Thank you for watching. Thanks for your patience. Hope this helps. Let me know if you have any questions. Take care.